welcome Shelly Stevens to give us her talk this evening. And it's about the gut and the emulsifiers. And Shelly is a toxicologist. She's from the US, as you probably know. And I'm going to ask her to introduce herself more. I believe she's the mother of somebody with a Cavernoma, rather than with a Cavernoma herself. And Shelly, it's over to you, and a very big welcome from us, and thank you very much for joining us.
Of course, thank you for inviting me. So thanks for the introduction. Today we are going to talk about the connections between the gut and cavernomas, and we're going to focus a lot on emulsifiers and talking about what emulsifiers are and what role they might play in, or what relationship they have with cavernomas. And hopefully I can answer some questions that you might have on this topic. So as you noted, let me see if I can get the slide to turn. There we go. So as you noted, I am a parent of a Cavernoma patient. So my daughter has a CCM lesion in the brainstem in her pons. And so I first became interested in this topic because as a parent, I felt like we really didn't have control over too many things. And I wanted to do research to see if there's anything I could do to help her live her healthiest life. And so I started reading the literature about cavernomas. And then the other reason I was interested in this topic is I work as a toxicologist. So I do human health risk assessment. And what that means is, for my job, I look at different chemicals that we're exposed to, either from the environment or in our food or water. And we review all of the data and literature about that particular chemical and determine whether or not it poses a risk to human health and at what doses or concentrations it might be a risk. And so although I don't specifically work with emulsifiers, I do kind of a similar process where I review literature about chemicals and look at how it affects human health. So for today's talk, I thought we would do a couple of introductions and then start talking about emulsifiers. So I'm sure most of you are already aware of what the gut microbiome is, but I just want to do a quick introduction and then just take a very broad overview of what the current scientific literature is that shows a link between the gut and CCMs. I think in your group, you might have gone over those studies before, so my review of those will be pretty quick. And then we'll talk about what a leaky gut is and what role emulsifiers and other food additives might have in creating a leaky gut. And then finally, we'll talk about eating an emulsifier-free diet. So we'll start at the beginning. And just to define what a gut microbiome is, because we're going to be talking about that quite a bit, your intestinal tract is lined with trillions of microorganisms, and most of these are bacteria. And these bacteria, these microorganisms, are what comprise your gut microbiome. So when we talk about the microbiome, we're talking about the organisms that are living inside your intestinal tract. And one thing that's really unique is that microbiomes are almost like a fingerprint. They are very unique to each individual. Although we all might have similar types of bacterial species in our guts, sometimes there are differences, and the ratios of those bacteria in your gut are very different from person to person. And a lot of it can be dependent on the types of food you eat or the environment that you're in. Things like stress or whether you're on antibiotics, all of those kinds of things can affect your gut microbiome. And I don't know about in the UK, but where I am, it seems like there's more and more companies talking about gut health and the microbiome, and there's all these products in the stores that say, eat this to have a healthy gut. And so I think that there's more interest in what the gut microbiome is, and a lot of people are starting to recognize that it might be important in overall human health. So the bacteria in our gut, sometimes you hear the word bacteria and you're like, oh, that's bad. It's actually a good thing. We live in symbiosis with the bacteria that are in our gut. And they do a lot of things that are helpful to us. For example, they help digest foods that we normally wouldn't be able to digest. They can produce vitamins and amino acids that our bodies can use for energy. They help metabolize therapeutics and drugs. They help train our immune system. And so the gut microbiome really has a broad influence over multiple body systems, which is kind of amazing. So how do these bacteria in our gut have this big influence? What do they do? Well, the bacteria in your gut produce small, let me just click through this to get the text. They produce small molecules. And these could be metabolites, or like I said, vitamins, amino acids, neurotransmitters. And these small molecules that are produced by the bacteria in your gut can exit the gut and enter into the circulatory system, and then have an influence over different body systems. And the bacteria in your gut can also interact with cells that are within your gut lining, like immune cells, I'll use my pointer here, immune cells or endocrine cells, and they can influence whether those cells produce different cytokines or hormones, and can help regulate what those cells produce and how it influences the rest of the body. So some of these molecules can reach the brain, and that's the key point here, because there is a definite gut-brain connection. The gut and the brain are in constant communication, and the way that they communicate is through these signals or small molecules from the gut that then reach the brain. And the types of signals that your gut sends to your brain is dependent on a couple of different things. One is the composition of your microbiome. So different types of bacteria may release different signals that can enter the circulatory system. And then also the overall health of your gut. So is your intestinal lining healthy and intact, or is it, we often use this term leaky, which we'll talk about more later. Those two main things can really influence what signals are going to go from the gut to the brain. So there's been a handful of studies over the past few years that have looked at a connection between the gut and CCM lesions. And as I mentioned earlier, I'm not going to go into a lot of detail on these, but I will just give you an overview of what these studies found, so that it helps kind of solidify in your mind that, yes, there actually is this connection that we should be paying attention to. So one of the first studies was published by Tang et al. In 2017. And it first started with something that they found by accident. So they had mice, they were studying CCMs in this mouse model, and they moved the mice from one facility to another. And they found that some mice were getting CCM lesions and some weren't. And they were really perplexed by this, and found that the mice that were developing CCM lesions had these gram-negative bacterial abscesses. So basically they had like an infection. And so they wanted to know what is going on. Why were mice with an abscess generating CCM lesions? And before I get into how they answered that question, I just want to define what gram-negative bacteria are, because we'll talk about them more often in this talk. Gram-negative just refers to types of bacteria that have a outer membrane compared to bacteria called gram-positive that don't have that outer membrane. And gram is just based off of a gram stain. So they stain these bacteria. Some take up the stain, some don't. So gram-negative versus gram-positive. But gram-negative bacteria, because of this outer membrane, they're often the ones that are more resistant to antibiotics, and they're usually the ones that are more pathogenic or might make you sick. And the other thing about these gram-negative bacteria is they have this molecule in their outer membrane called lipopolysaccharide, or LPS. And we're going to be talking a lot about LPS in this talk. All right, so back to the lab where they found mice with abscesses that had CCM lesions. They wanted to do an experiment to kind of test out what this relationship might be. So they took their CCM mouse model. Now, these were healthy mice, not ones with the abscess. And they injected the mice with gram-negative bacteria or to that LPS molecule by itself. And then they compared mice that were injected to ones that were a control. So they weren't exposed to these things. And they looked at whether or not there was any difference in CCM lesion formation between the controls and those that were exposed. And they found that mice that were injected with the gram-negative bacteria, or TLPS, had an accelerated an acceleration in CCM lesion formation. So that was the first clue that there is some link between bacteria that's normally found in our intestinal tract and CCM formation. And then there was another study by Detter et al. In 2020, and they took mice who already had CCM lesions, and they sort of did a similar thing. They injected them with LPS to see what would happen, compared them to controls. And they found that in these mice, those that were injected with LPS, there was an increase in bleeding in the existing lesions. It was a very small group of mice that they did this in, and probably some follow-up studies are needed, but it again shows some sort of relationship between this molecule produced by bacteria and CCM lesions. So how is this working? How does this LPS molecule induce lesions and cause bleeding in mice? To answer this question, it's good to just take a quick step back to look at what we know about Cavernoma formation. And of course, the formation of lesions is really complex, and we are always having new studies that tell us more information about how these form. So I'm just going to focus on the one pathway that's relevant to this LPS molecule. So we'll go through that. This is a cartoon of a blood vessel in your brain. Your blood vessels are lined by endothelial cells. And in these cells, you have the CCM proteins. And I'm sure all of you are aware of CCM1, CCM2, and CCM3. The CCM1 also is called CRIT1, and CCM3 is also called PDCD10. These proteins inside your endothelial cells will form a complex. And one of the functions of this complex is to inhibit a pathway, a signaling pathway, called the MEKK3 KLF24 signaling pathway. I know that's a lot of acronyms. I apologize. But the key thing to remember is that the function of the complex is to inhibit this signaling pathway. And if you have a mutation in one of those genes that disrupts the function of this complex, you can get an overstimulation of this MEKK3 signaling pathway. And when this pathway is overstimulated, that can promote the formation of CCM lesions. So where does LPS come into play with all of this? It turns out that LPS can bind to a receptor on the surface of your endothelial cells called the TRL4 receptor. And when it binds to this receptor, it can activate that same MEKK3 signaling pathway. So you can imagine if you have a mutation in one of your CCM genes, so you already have some stimulation of this pathway, and then on top of that, you add LPS that is further activating this pathway. And again, when the pathway is overactivated, you can get formation of CCM lesions. So here we can see that you have gram-negative bacteria that have this LPS molecule. They're in your gut naturally. And under certain circumstances, that LPS molecule or the gram-negative bacteria can escape the gut, enter into the circulatory system, and travel up to the brain. And then you can get activation of this TRL4 receptor and stimulation of this pathway. Another one of the studies that was done by Polster et al., they decided to look at the differences in the gut microbiomes between patients who had CCM lesions or CCM mutations and those that didn't. And they found that CCM patients had distinct microbiomes compared to non-Cavernoma patients, and that these microbiomes were enriched in the amount of gram-negative bacteria that they had. And if you recall, gram-negative bacteria are the ones that produce this LPS molecule. And in addition to having more gram-negative bacteria, there was an abundance of genes that are known to help synthesize this LPS molecule. And so already we're starting to show a distinction between the gut microbiomes of CCM patients versus a healthy population. So just to sum up what we know that gram-negative bacteria and LPS can accelerate CCM formation and bleeding in mice. We know that gram-negative bacteria are found in our gut microbiomes just as part of the microbiome. But CCM patients have more of this gram-negative bacteria and LPS in their gut microbiomes compared to non-CCM patients. And then we talked about how the makeup of the microbiome can influence what signals are sent to the brain. Right. So the other part of this story is the health of the gut and the whole idea of having a leaky gut. So what does that look like and what does it mean? Normally, in a healthy gut, so actually, if we take a look at this cartoon here, at the top here, this would be the inside of your gut where the microbiome is. And then you have your gut lining, which is lined with endothelial cells. And then you have your circulatory system on the other side. So in a healthy gut, you will have endothelial cells that are very tightly packed together. And they have these structures called tight junctions in between the cells that help hold them together. And this is a really cool image. This is an electron micrograph of two different cells. So you have one cell on the left and one cell on the right. And these junctions where you can see it coming together, those are the tight junctions. So they really act as a barrier and also a regulator of what can pass between the inside of your gut to the outside of your gut into the circulatory system. The other aspect of a healthy gut is this mucosal barrier. So this is shown in two different layers in blue. And the mucosal barrier, you can almost, similar to the mucus in your nose, it acts to flush out or an extra barrier to pathogens. And so this mucosal barrier also can prevent things get from the inside of your gut getting out. What happens when these things aren't there? So we talked about the healthy gut where you have the tight junctions, you have a nice thick mucosal barrier. When you hear the term leaky gut syndrome, it's normally talking about cases where there's some sort of faulty tight junctions. So maybe these endothelial cells aren't tightly packed together like they would be, and there's a freer passage for molecules to get from the inside into the circulatory system. Or it might be referring to a compromise in that mucosal barrier. Maybe it's not as thick, or maybe there are areas where it's just gone. So whenever you hear the term leaky gut, this is what it's talking about. It's talking about defects in these barriers that help separate the inside of the gut lining and your circulatory system. So there was a third, well, I don't know how many studies we're on now, maybe 4. There was another study where they were really focused on looking at the CCM3 mutation, because we know that patients with CCM3 mutations, especially the familial one, usually have more CCM lesions, and they often appear sooner than the CCM1 or CCM2 mutations. And they wanted to know if there was any relationship between the CCM3 mutation and the gut. And one of the reasons they were looking at this is because the CCM3 gene, it's not only important in the brain, it also plays a role in the gut. And it helps the cells in the gut produce this mucus layer. So it is important for the formation of this mucosal barrier that we've been talking about. So they looked at the mucosal barrier of mice with familial CCM3 mutations, and they found that the mucosal barrier indeed was compromised in these mice. And these mice also had symptoms of colitis and an increase in this LPS molecule circulating in their blood. And so the increase in CCM formation in mice that have this mutation seemed to be related to the compromise in that gut mucosal barrier and the increase in LPS. So what about other things that might compromise the gut? I mean, other than CCM3. They did another study, this was by Tang et al. In 2019, where they induced colitis in some of their CCM mouse models. So they used a chemical that is known to induce colitis or inflammation in the gut. And they found that when they injected the mice with this chemical that induces inflammation, they saw an increase in lesion volume in these mice. But the next question was, well, what about dietary agents? Because, I mean, humans are not going to be injected with something that induces colitis. But what about dietary agents or food additives that are known to impact the gut? And specifically, they were looking at emulsifiers, because emulsifiers have a long history of being disruptive to the gut, specifically certain ones. And so studies that are looking at the gut often use emulsifiers to kind of cause harm in their studies as a control. So they took mice and they fed them diets for several weeks that contained an emulsifier called polysorbate 80 or P80. And then they looked to see what effect this diet had on the mucosal layer or mucosal barrier in these mice. And it turns out that the the thickness of the mucosal barrier in mice that were given emulsifiers was much thinner than those that were the control mice. And so feeding the mice the polysorbate 80 caused a leaky gut type symptom. And in addition, these mice also showed a significant increase in the formation of CCM lesions. So again, we're seeing this connection between a leaky gut and formation of CCM lesions. And so just to sum up what we know and re-review, we know gram-negative bacteria and LPS accelerated CCM lesion formations and bleeding in mice, that these bacteria normally found in our guts, but CCM patients had more gram-negative bacteria and LPS compared to non-CCM patients. And those with the CCM3 mutation, familial CCM3 mutation, also had an additional compromise to the mucosal barrier in their guts. And that a leaky or compromised gut caused by either chemicals that induce colitis or feeding mice emulsifiers was associated with CCM lesion formation. And so the conclusion of all of these studies was, at least in our group at the US Cavernoma Alliance, was the recommendation to try to avoid emulsifiers in your diet because of this gut CCM connection and what we know about emulsifiers so far. And so the main take-home message was we really want to support a healthy gut. We want to try to prevent a leaky gut as much as possible. And that's a great idea, but there wasn't a lot of guidance as to how one can go about doing that. Because there's so many things that influence gut health. How stressed you are, your genetics. For, some people have celiac, or some people have sensitivities to soy or other various dietary allergies or sensitivities. The environment that you live in can influence your gut health. If you're on medications, for example, antibiotics can have a significant impact on your gut microbiome, your age, and then of course, diet. And if you were to Google diet and gut health, you would get all this information about artificial sweeteners and sugar and high-fat diets and also emulsifiers. So there's many, many things that go into gut health, and we're obviously just going to focus on emulsifiers right now, but I just wanted to highlight this, that it's not just emulsifiers that can impact your gut health. So what are emulsifiers? I'll define what they are, and then we'll talk about whether or not all emulsifiers are bad. Are they all the same? What do we know about them? And then how can we go about avoiding them in your diet if you choose to do that? Emulsifier is really just an umbrella term. It's a term for any compound that allows two immiscible phases to mix. So something that it will, like oil and water, it will help them mix. And so emulsifiers are added to food to help stabilize it. It can be used as a gelling agent, as a thickener, and really helps keep foods having the consistency that they're supposed to have. Mostly emulsifiers are found in processed foods, but when you think about processed foods, at least the first thing that comes to my mind is, unhealthy foods. But there's a lot of healthy foods and food staples like bread and yogurts and maybe non-dairy milks that are not really unhealthy, but they contain emulsifiers in a lot of cases. There are also foods that have natural emulsifiers in them. And we can talk about that more, especially during the question and answer phase. I'm going to focus today on food additives. So when they specifically add emulsifiers as an ingredient into foods rather than ones that are naturally occurring. But I'm happy to discuss the other later. How many emulsifiers are there? Well, that's a big question. The Food and Drug Administration classifies 171 food additives as emulsifiers. And I looked up for you guys that the European Food and Safety Administration classifies 65 food additives as emulsifiers, but then they have another 158 that are classified as other, and anti-caking agents and thickening agents were in that list. And those also tend to have emulsifier capabilities. So it's a little bit hard to say. And just because the FDA classifies one as an emulsifier, the EFSA may not. So it's a bit complicated. I did put the link for the EFSA classification list on here. And hopefully if this talk is posted, you'll be able to go to that link and just easily find the list of emulsifiers that are classified in the UK. So I mentioned that emulsifiers is this big umbrella term. So how do we know which ones are bad? And how do we define what's bad? I want to point out first that all of the emulsifiers that are classified as such have been deemed safe by the FDA and the EFSA. So these organizations have already reviewed these food additives and determined that they're safe for use in food. However, I will point out that when the EFSA reviews an emulsifier or any food additive, they are not looking at whether or not those food additives have an impact on the gut. They are looking more for more serious signs of toxicity. So do these food additives cause behavioral changes in animals? Do they cause death? Do they cause severe changes to major organs like the liver or the kidney, or severe weight loss? So they're looking at more traditional signs of toxicity in experiments, and they're not looking at more subtle changes in the gut microbiome. And in most cases, when companies do toxicity assays on compounds or chemicals they produce, they don't do a real close look at the gut. They might look at the intestinal tissue under a microscope, but they're not going to check whether it causes a leaky gut, for example, or anything like that. So I just wanted to point that out. So what I wanted to do was kind of mimic what I do for my normal job and collect all of the studies that I could find on emulsifiers and the gut and record what the key findings were in those studies. And then I came up with a scoring criteria, and I'm going to define all of that in a few slides from now. But in reviewing all of the studies and using my scoring criteria, I then tried to give each emulsifier a score, and the scores would reflect whether or not the scientific data shows if that emulsifier has a negative effect on the gut, or maybe a beneficial one, or maybe we're not quite certain at this time. So just to walk through a little bit more of what I did and what approach I took, I first wanted to define what kind of data we have on emulsifiers. And really there's 3 streams of data. And the first is what I will refer to as in vitro studies. So these are studies that are done in a test tube or a petri dish. And they might take bacteria or human fecal samples, for example, and add emulsifiers to this bacterial culture and look to see whether the emulsifier affects the survival of the bacteria or if it affects the production of LPS by the bacteria in the test tube. And it's important to note that studies done in test tubes or in vitro are often not necessarily translatable to what we see in humans. And so I would say that data using this evidence stream is kind of the weakest data, and it's more supportive than definitive. Another type of data we see are animal studies, and we've already talked about some of those where they expose mice or rats to emulsifiers. They look for changes in the microbiomes of those animals. They might look for production of LPS or inflammation, changes in that mucosal barrier, or correlations to disease, like correlations with the formation of CCM lesions. And studies in animals are traditionally used in toxicology. And although animals are not people, we do have quite a bit of knowledge about what systems are similar and how we can go about using the data in animals to apply to humans. And then, of course, the third data stream is studies in people, and there are very few of these. Sometimes they're intervention studies where they might have humans, populations eat specific diets. So maybe they'll have a group eat an emulsifier-free diet versus a normal Western diet. Do we want to mute? I think we're getting some background noise. Anyway, there's very few of these human studies, but we can look at differences in microbiomes between humans, like they did between the patients with CCM lesions versus the ones without. You can look at levels of EPS and LPS in circulation, and you can look for correlations to disease. All right. So I have to apologize. This study is a few years old, so the numbers aren't necessarily relevant, but it's just a tree scheme of what I did. So let's say that I identified 180 studies. These studies were identified by doing keyword searches on PubMed and Google Scholar and some other databases. I unfortunately no longer work at a university, so I don't have free access to all scientific studies anymore, which is really unfortunate. But I found some of those studies had available PDFs of the full study that I could look at, some didn't. Out of the studies that had the full PDFs, I separated them into the different categories of data streams, and then looked at what effects they showed and if there was something negative or not. And then all of those studies that I couldn't really look at, I took a look at the abstracts, and I kind of just have these studies banked for another time. I can try to contact the authors to see if they'll send me the study. But for now, those ones that I couldn't fully look at aren't really included. And then I created a database. I know this is very small. It's not important for you to read all of it, but basically it's just a very long Excel spreadsheet that has what emulsifiers the study used, what model system the study used, what the effects were, and then the references and the links to those references. And this is just a small snapshot of a very long list that I have. And my scoring criteria was probably the most difficult thing to come up with. And of course, this is just based on my experience and opinion, and others may have done this differently. But for me, if in order for me to score an emulsifier as having a negative effect, I wanted to see evidence from multiple studies, primarily in animals or humans, because those are more reliable, that showed a negative impact. And a negative impact I defined by showing an increase in gram-negative bacteria, or an LPS, or inflammation, or maybe the studies showed a compromised mucosal barrier or gut permeability. And then there's groups of emulsifiers where we just don't have sufficient data. There might be mixed results, or there might be only data from that lower quality in vitro type study. And so those I have currently scored as a caution, because we don't know one way or the other. And then, believe it or not, some emulsifiers actually have a beneficial effect on the gut. And I scored these emulsifiers as beneficial if there were multiple studies in animals or humans that showed some sort of benefit. And by benefit, meaning there was a positive influence on the gut microbiome, or the types of bacteria in the gut, or maybe there was a stabilization of the mucosal barrier in that case. And then, unfortunately, there's lots of emulsifiers where there just is no data at all. I can't find any studies on them. So we don't know what to do with those. Then I created this emulsifier scorecard, and I'm just going to flip through these. So I have them all here. And these, it's not complete. There's still a lot more to do. I have alerts set up on my email, so every time there's a study posted on emulsifier and the gut, I get an alert and I can review it and add it to my database. But I haven't actively looked for about a year. Although if I get alerts, I will put those on. The thing I want to point out is that this scoring that I came up with doesn't tell you a lot of things. It doesn't tell you whether those studies are of good quality. So one of the things I do for my normal job is, in addition to reviewing the science, the literature that's available, we also review the quality of those studies. Did they have proper controls? Were they using a sufficient sample size? That takes a lot of time. And so it's not something that I did in this case. It also doesn't include studies that I missed. Another thing that I do for my job is something called systematic review. So we have librarians who work with us, and they will retrieve every study from, say, 1900 to 2023 that mentions the name of the chemical you're looking at. And we will get thousands, and we go through all of them. In this case, I was just using keyword searches. Although I have a pretty good knack for doing keyword searches because I do it often, there's inevitably going to be studies that I missed that weren't included. And then the last thing that this scoring system doesn't tell you is whether or not the doses or concentrations used in those studies were relevant. So maybe they feed mice an emulsifier, but the amount that they feed them is so ridiculously high that humans in their diet, even if they eat terribly, would never get the same level, be exposed to the same level. And so this is something that can happen all the time in animal studies where they use really, really high concentrations of things. And so I didn't take that into account, and that certainly can have an effect on the results. So with all of that information, how can you go about eating an emulsifier-free diet? And I just want to share with you what we've done, or what I've helped the Angioma Alliance group do in the US. So the first thing is to make a decision. You know, you've got all this background information now. You've been exposed to the few papers that were published showing the connection between the gut and cavernomas. You have an idea of what emulsifiers are and how you can find lists of them. And you just have to decide, is there enough evidence that you've now seen that makes you want to avoid emulsifiers? And maybe you want to generally try to avoid them, but if you go out to eat, or you have a special meal at a holiday and you happen to have something with emulsifiers in it, you're not going to stress about it. Or maybe you've looked at all of this information and you're like, I really just want to avoid those that have scientific data showing that they have a negative effect, because clearly not all emulsifiers are the same. Then there's also groups of people who say, I just want to completely avoid emulsifiers altogether. And then also people who take a look at all of this information and are like, I'm not convinced, I don't want to modify my diet. So you have to make a decision of what's right for you. And then the next step is to start looking at food labels. Well, maybe the step before that is to print out the list of emulsifiers from the EFSA website that I put up there so that you know what you're looking for. And then you start to look at food labels. And for example, this is from, I think, some almond milk, and it has the emulsifier carrageenan in it. And actually carrageenan is one of the bad ones and is often used to purposefully induce intestinal effects in animal studies as a positive control. So you want to start looking at food labels. And one thing that we did with the Angioma Alliance is we kind of crowdsourced this. So we worked as a group to put together a list of emulsifier-free foods. And I'm just going to show the next slide quickly. And we have a list of it on our website. And what we did was we had members, when they went to the grocery store, if they found a food product that was emulsifier-free, they would email it to me. And then we created this list, and it's broken down by food type. So what's shown here is the bread products. But we also have dairy, specifically like yogurt and milk alternatives and snacks and so on and so forth. So on this list, we included a picture because when you go to the grocery store, just a name isn't always helpful. And the category type and a link to where you can find the ingredients of those specific products. So I will say that this was very helpful when we first did it to a lot of people, and especially when many of us were working on it together to form it. I don't know how often it's used now. And we did try to create a website where people could just import their information, and then I could auto-populate this list, but nobody has actually used it. So, it all depends on how motivated you guys are to try to come up with this list. But once it's done, I think it's very, very helpful. And unfortunately, a lot of the things that we have on our list, you probably have different products in the UK, so it may not be helpful. Another thing that we do is we have a CCM healthy cookbook. So, and these are all emulsifier-free meals that people submit. And once a year, we collect more recipes, we put them together. and have a cookbook for everyone in our group to use. And then the last thing is something called the FIG, or the Food is Good app. And I don't believe this app is currently available in the UK, but they are planning on making it available soon. So I worked with the CEO of this company and just volunteer. I have no like stake in them or anything. I'm not trying to promote, but just to let you know that it may be available. But what I did was I helped them create an emulsifier-free diet for Cavernomas within their app. And if you download their app, then you can click on this particular diet. And what it does is it has all of the products in a grocery store, and you can say, find products that are emulsifier-free, and then it'll give you a whole list. Or if you're looking for a particular type of product, you can search, and it'll only give you the ones that don't have whatever ingredient you say you don't want. It's kind of very useful. And that's kind of where we are right now. We have people who utilize this app. We have people who utilize our food list that we keep adding to. And my family, we eat an emulsifier-free diet for the most part, although some of the ones that I've found to be beneficial in my searching, I don't really worry about too much.